If the sea was like a big, well-mixed bathtub, we could know everything about it by analyzing only 1 cup of water. Living in that cup of water are plants, called phytoplankton, and animals, called zooplankton, often smaller than the eye can see. We call them plankton, which comes from the Greek word for wandering. It means that the creatures cannot swim against the current. They must go with the flow. But even though they're small, these little creatures are very important. They form the base of many food chains. But the sea, of course, is not like a well-mixed bathtub. How do we know that? Well, since 1931, ships have been recording plankton between 2 sheets of silk. The continuous plankton recorder is continuously towed behind a ship. This can be a research vessel or a commercial vessel. Each plankton individual on the sheet is eventually identified under a microscope in the lab by an expert. From this type of research we know, for example, that some warm water plankton species have moved almost 1,000 kilometers northwards over the past 50 years. We can also catch plankton in nets that are pulled up through the water column. This technique was already used in the 17th century, for example by Darwin. The nets sample plankton from some 100 meters of water or more and can be closed at different depths. From research with plankton nets, we know that many zooplankton species feed in the top 20 meters of the water column during the night and then migrate down during the day to avoid predation. The spatial resolution of the plankton net is higher than with the plankton recorder, but from a new technique of plankton sampling, we know that plankton nets can still miss some patterns in plankton. This new technique is the plankton scanner. It's an underwater microscope that is towed behind a ship and takes photos of plankton continuously. It is typically employed together with a CTD, a device that measures conductivity or salinity, temperature and depth. Research with the underwater microscope has revealed an interesting pattern in the most abundant multicelled organism on our planet. Sometimes in summer, the deepest layer of the water can become depleted in oxygen. This is called anoxia. Like all zooplankton, copepods need oxygen. So in the case of anoxia, they move up in the water column. But this is risky because higher in the water column there are more predators. Scanning these communities with the underwater microscope has shown that Euthona individuals carrying eggs are slightly more risk-averse than individuals that are not with babies. They still move up into water with oxygen, but they stay slightly lower. At the Royal Dutch Institute for Sea Research, researchers have already gathered 500 million images of plankton, and there's many more images to come. When their high-performance computers have processed this heap of information, they hope to reveal many more patterns in plankton. And in the meantime, we marvel with them at the beauty of one of the most important groups of organisms on our planet.